we have the power to look at the genetic blueprint of life, identify the smallest glitches in our DNA responsible for life-altering diseases, and develop tailor-made drugs to fix them. What was once science fiction is now groundbreaking reality, thanks to genomic approaches to drug discovery. But how does the genetic code translate into life-saving medications? How do we turn complex data from DNA sequencing into validated drug targets? And more importantly, how can you, as a researcher or enthusiast, understand and harness this process? In today's video, we are diving deep into from genes to drugs. By the end of this video, you will understand how cutting-edge genomic technologies are revolutionizing target identification and validation, the very backbone of modern drug discovery. Let's start our journey through the fascinating world of bioinformatics, where genes transformed into cures. Make sure to stick around till the end. This content could fundamentally change how you see drug development and disease treatment. Before we dissect how genomics is shaping drug discovery, let's step back and look at the big picture. Drug discovery has two main phases. Number one, target identification, and number two, target validation. Target identification. This is the what biological element, gene, protein, pathway is responsible for a disease, and is it actionable? Next, target validation. This is the why. Why is the target relevant for therapeutic intervention, and can it be modulated effectively with a drug? What genomics allows us to do is unlock the hidden instructions encoded in DNA. These instructions reveal the blueprint of genes and proteins involved in both health and disease. With this roadmap, researchers can find the exact gene responsible for malfunctioning cellular mechanisms and determine interventions to restore balance. Genomics has minimized guesswork and maximized precision, and that's where bioinformatics becomes our weapon of choice. Tools like CRISPR, gene editing, RNA sequencing, and genome-wide association studies have opened a treasure chest of possible targets. But this target chest requires systematic digging, interpretation, and validation through bioinformatics. So how exactly do we move from raw genomic data to real actionable drug target? Step 1 is genome-wide association study. PAUSE is your starting point for identifying genetic variations linked to specific diseases. Think of PAUSE as scanning a giant crowd of genomes for genetic commonalities among people with similar diseases. For example, by comparing DNA sequences from people with type 2 diabetes to those without, researchers can identify single nucleotide polymorphisms associated with the condition. These SNPs point to potential suspect genes that might be driving the disease. Step 2, functional genomics. It's not enough to know where the problem lies. You need to know what it's doing. That's where functional genomics comes into play, analyzing how specific genes express themselves. Tools like RNA sequence reveal patterns of gene upregulation or downregulation in disease states versus healthy states. So we need to explore the gene's interactions with protein, network, and metabolomics. Step 3, big data bioinformatic pipeline. This is where bioinformatic tools like BLAST, string, and CAG pathways shine. By leveraging advanced databases, we map how genes interact with other proteins, signaling networks, and downstream biological processes. The result, you not only spot candidate genes but also understand how they fit into the system. It's incredible. Now that we have a potential target, how do we validate it? So target validation is about proving why our chosen gene, protein, pathway is truly the culprit and can be drugged. Here is how it's done. So step 1 is experimental validation. Laboratory techniques like CRISPR allow researchers to edit or knock out the suspect gene in a controlled environment. For example, knocking out gene X should prevent cancer cells from growing. Not, gene X might not be the true target. So step 2, transcriptomics and proteomics. Single experiments aren't enough. To double check using transcriptomics to see if the gene slicing changes other pathways, and proteomics validates whether the downstream proteins this gene regulates get effective, further cementing its role in disease progression. Step 3, model systems. Use animal or cell culture models to study how the druggable target behaves in a biological system. These models simulate how successful therapeutic intervention might look. So what does all this lab research talk translate into real-world therapeutics? Number 1, gene therapy. Using genomic techniques, diseases once thought incurable are now manageable with precision prevention intervention. For example, spinal muscular atrophy is treated with gene-based drugs like Zolgensma. Number 2, cancer immunotherapy. Leveraging gene profiling for personalized treatment, drugs like CAR-T are built off genomic information from individual cancer patients. Number 3, rare diseases drug development. Genomic studies have allowed pharma companies to develop orphan drugs targeting diseases caused by rare genetic variants. What about the intersection of AI and genomics? The process of drug discovery has shifted from manual data handling to integrating machine learning models capable of predicting novel drug targets with unparalleled speed, modeling folding patterns of proteins, for example AlphaFold, conducting virtual drug screening to test thousands of compounds against gene targets, saving time, money and resources. The pharmaceutical industry has already begun implementing these models in their pipelines. Are we heading towards AI-led biotech dominance? Let us know your thoughts in the comments. To summarize, genomics enables precision in identifying drug targets. Functional genomics and bioinformatics tools validate these targets for real-world applications. Next, AI pushes the boundaries of what's possible in drug discovery by accelerating every aspect of the pipeline. If you found this exploration of genes to drugs fascinating, I have got lots more coming your way. So make sure you subscribe and hit that notification bell. Now, here's a twist. There's a question for you. So what do you think is the most exciting advancement in bioinformatics today? Drop your thoughts in the comments below.